Hi Biology, this is Miss Adderley. I'm going to make a video to explain tomorrow's assignment, especially for students that are remote learning or quarantined right now. So it'll be under the things to do in the classwork tab, cell structure exploration lab, and I'm going to explain what this is. This will be due on Monday and you can work on this tomorrow in class. But it is an activity to look at different types of cells. So you're going to click on this. There's two documents or two things that go with it. First off, you have the cell structure lab itself that has uh, the directions with it, and it also has 15 pictures of different organisms. And then the other thing you need to open up is the cell structure exploration lab itself. And I'm going to go over both of those really quick. So cell structure lab, the directions. What we're going to be doing for each one of these structures is to determine if it's a prokaryote or eukaryote. Prokaryotes don't have a nucleus. Eukaryotes do have a nucleus. We're going to see if they're unicellular or multicellular, and that is easy to determine based on the picture itself. So if you see cells that are side by side, it's usually a multicellular organism. If there's space in between, or if there's a single cell itself, it'll be unicellular. We're also going to determine the specific type of cell. So we have animal, plant, fungus, protist, eubacteria, or archaebacteria. Those are the different things we might see. You need to determine if it's a nucleus or no nucleus. Well, if you pick prokaryote, then you know it doesn't have a nucleus, and if it's a eukaryote, it does. And then the last thing is the cell wall or no cell wall. If there's no cell wall, you actually do not have to answer F, which on the assignment itself it'll be number 6. If there is a cell wall, I want you to determine the type of carbohydrate in the cell wall. So plants have cellulose, fungus have chitin, eubacteria has something called peptoglycan, archaea has an S layer, and diatoms, which are actually a type of algae, have silica cell walls. I'm not going to go over this too much with you guys, but I'm going to show you each of the pictures. This is a mushroom. A couple things you can do. I can see by this picture there are multiple cells next to each other. So that tells me it's a multicellular organism. They also, most of the cells seem to be a little more box shaped, and they have kind of a thick boundary around them, which might indicate there's a cell wall. Mushrooms do have a cell wall because they're a fungus. So those are things you'll be doing. Streptococcus, you can look up these organisms too if you have trouble with it. But this is the next one. It's a type of bacteria. Onion, we know what onions are. Spirilla, amoeba. So on the amoeba, I do want to point out that it does not have a rigid structure. It has a lot of things coming out of it. Those are also actually called pseudopodia, which means false feet. It's very movable, and so that definitely tells me there's no cell wall, which means it has to be an animal cell. Algae. This is just a typical fungus cell again. But if you look at the difference between this one and this one, this is an animal cell, so I just tell you what it is because it's just a generic animal cell. The boundaries of the cells are different shapes. They look a little thinner. If you look over here, some of those cells have really thick coloring around them, and that's kind of indicating that there's more of a cell wall. A paramecium. The paramecium is not this big blob of dark green. It's actually the organism right there. So you can see there's only one cell to it. Paramecium have a lot of cilia, which you actually can see in the picture. 10 is E. coli. E. coli is a type of bacteria. Diatoms, which I said are algae. So this is actually the skeleton of diatoms. Diatoms are actually used in like forensic studies. So say they find a victim that's in a particular area. Sometimes they study the types of diatoms on their shoes if to see if they actually died in that area or they were moved. So that's just kind of a fun, interesting fact with diatoms. Archaebacteria. RK bacteria are, there's two types of bacteria, RK and U bacteria. They're very different, but they do look similar. Euglena is the next one. Euglena is actually a single-celled animal cell. It has a flagellum at the front, and then there's an eye spot, which is what that dark red structure is. Corn, we all know what corn is. This is actually a picture of the different stages of mitosis, which we'll get into next semester. And last one is a eubacteria mix. So there's different shapes. We have some what looks like streptococcus, but there's a lot of different circular bacteria. And then there's the rod bacteria in there as well. So those are different types. So this is the PowerPoint that you're going to use to look at those different cells. So you'll go back and forth. So in this lab, you'll examine 15 different microscope slides to determine the type of specimen that you're looking at. Most of the information can be determined by simply looking at the slides themselves, but some material may have to be looked up online. For each microscope picture, you will have to determine the following information below. I provided you with an example. Please follow the pattern. So this is the example. All I have done is the thing that you need to type in is the name up the top next to name of specimen. And I have that directly on the slide for you. And everything else, you're just going to highlight the answer that you think is right. So mold is the type of fungus. So I highlighted fungus. Fungus has chitin in it. It has a cell wall. It has a nucleus. It's multicellular and a eukaryote. So you're going to be highlighting the information opposed to typing everything out. There's 15 sections already ready for you that correspond with the pictures that are on the cell structure Google Slides. You can delete the type of cell wall, the one that isn't appropriate, the type of carbohydrate. So if I wanted to, I could just put cell wall and get rid of A, C, D, and E and just have fungus because it is a type of fungus. But what I do want you to do is if it's an animal cell, please delete number 6 entirely. So you will just go and highlight it like this and get rid of it because it's not necessary, because animal cells don't have a cell wall. So again, you have 15 sections already set up like this. What you need to do is at the top, you're going to type in, the first one was a mushroom. So a mushroom cell, so I'll type that in, and then I want you to highlight it. I prefer yellow, but you really can highlight the color that you choose. So a mushroom cell, and then I would go through and highlight the appropriate things. You'll do that for each slide. I do have the number on that for each one. And so each one has its own separate section already ready for you to do. This should be pretty straightforward. Of course, you can use Google search for anything that you're unsure about. You can always ask me tomorrow in class if you're working on this, or if you're working on it at home. Send me a private message, email me. I'll be more than happy to help you out if I can. All right, so this is the instructions. This will be the assignment that will open up tomorrow. It's scheduled to open at 7:50. And this will be your assignment for tomorrow. If you have any questions, please let me know. Thanks.